Hello everyone, welcome back to my channel, IGCSE Biology Help. In today's video, I'm going to be solving the Biology Paper 4, Extended, May-June 2023, Paper 4, Variant 1. Make sure to subscribe to my channel and like my videos to receive more content like this. Let's start solving. Question number 1a. Figure 1.1 is a diagram of the digestive system. Each letter may be used once, more than once, or not at all. State the letter of the part shown in figure 1.1. So the part that produces bile is actually the liver, which is K over here. So you're going to write down K. And the part that produces gastric juice, this is actually C over here, which is the stomach. And the part that produces urea is again the liver. While where maltose is digested, this is actually H, which is the small intestine, and where trypsin acts is also in the small intestine over here. So this was question 1a. Now question B, 1b says, a student investigated the effect of bile on the digestion of fat in milk. They set up 3 different test tubes. Test tube A contained milk and bile. Test tube B contained milk and lipase. Test tube C contained milk, lipase, and bile. They used an indicator that is pink in alkaline conditions and colorless in acidic conditions. So if the solution suddenly turns alkaline, then it will go from colorless to pink, for example. And if it goes from alkaline to acidic, then it will go from pink to colorless. They added the same volume of indicator in each test tube. The student observed and recorded the color of the contents of each test tube at 0 minutes, 20 minutes, and 40 minutes. Table 1.1 shows the results of the investigation. So over here in test tube 1 or A, we can see that at 0 minutes it was pink. That means that it was alkaline. And at 20 minutes it remained pink until 40 minutes. So the whole test tube A was alkaline. It remained alkaline because it only contained milk and bile. Now B, it turned from pink. It was remaining pink from 0 minutes until 20 minutes. And then at 40 minutes, it suddenly turned colorless, which means it became acidic. So this was the test tube that had milk and lipase over here. So also test tube C contained milk, lipase, and bile. So it contained the 3 ingredients. Pink, it was pink from 0 minutes, and then in 20 minutes and until 40 minutes it became colorless. So over here we can see that in B it was taking more time to become acidic. Unlike test tube C turned acidic, which is colorless, in less than 20 minutes, but B actually took more than 20 minutes to become colorless, which indicates that it was acidic. Now let's see the question. I says explain the results for test tubes B and C in table 1.1. So you have to say why there was a change. First of all, we can see that the pH decreases as fatty acids are produced. Lipase breaks down fat and fatty acids produced faster in test tube C than B. So like I said, test tube C was actually taking less time than B to become acidic because it has lipase, which breaks down the fatty acids faster because it's an enzyme, and bile emulsifies fat and breaks down fat into smaller particles, and the bile increases surface area for action of lipase. So this is what you should say, why there is a difference between the results in B and C. Double I says explain the purpose of test tube A in table 1.1. So this one only contained the milk and bile. So this is actually a control experiment, which means it is used to actually compare the results from B and C and show like a better perspective of the comparison and show like more accurate results. Demonstrate that bile does not digest fat and does not make solutions acidic. So of course we know that bile it just breaks down the fat from large molecules and to smaller molecules, so there is no really anything inside the bile that can make the solution become acidic. And also it shows that the enzyme lipase is required for fat breakdown because the solution didn't turn acidic unless it had lipase. So this is the use of the control experiment to just prove a point. Now C says the action of lipase is affected by temperature. Figure 1.2 shows the axis for a graph of the effect of temperature on the activity of lipase. Complete the graph by drawing a line to show the expected effect of temperature on the activity of lipase, and adding a label line and a label to show the point at which all the lipase has been denatured. So over here we can see I drew this graph. You have to draw it yourself. So I drew that the enzyme activity, as the temperature was increasing, the enzyme activity was increasing until this point over here. And then it started to decrease until it reached to 0 over here. The enzyme activity was 0. This is because the lipase denatured, and I labeled that over here. I wrote that down. You have to also write that down in your answer. So when the lipase denatured, this means, what does denature mean? Denature mean that, for example, if your enzyme looked like this, for example, let's just say it looked like this. When the temperature increases, it's like melting. It melted and the shape changed because of the different temperature or the pH conditions. If it's too high, the temperature, then the shape will actually change. It may be something like this. It changed. It wasn't like before. As we can see, it was like a square over here, and then after the temperature increased, became like that. So this means denatured. So if there is a square, a substrate that wants to bind, for example, sorry guys, my drawing is really bad. Over here, for example, if you have something like this that wants to bind, and if it comes over here, then it will not know how to bind because there is a different shape. So this is what means denatured. And I also explained this in my complete syllabus video if you want to check it out. Now, D says, explain why lipase cannot be used to catalyze the breakdown of proteins. Lipase enzyme is only specific to substrate of fat or lipids. So, of course, you cannot use lipase for proteins because lipase is for lipids, which is fats or oils. So you cannot use that to break down protein. It's something else. Proteins need another enzyme. Protein molecules will not fit into the active site of lipase as the shape of the protein is not complementary. It's not the same thing. Protein cannot bind and cannot form enzyme substrate complex. So this is the problem with why you cannot just use any enzyme. The enzyme has to be complementary to the active site. Like the substrate has to be complementary to the shape of the active site so that they can bind and they can actually form the enzyme substrate complex, and it can catalyze the reaction. So this is why. Now for question 2a, a student investigated osmosis in potato plant cells. He immersed cubes of potato tissue in water and different concentrations of sucrose solutions for 30 minutes. The masses of the potato cubes were measured before and after immersion. The percentage changes in the mass were calculated. Table 2.1 shows the results. Over here in I, it says, using the information in table 2.1, calculate the percentage change in the mass at 1 mole decimeter negative cube. Give your answer to 2 decimal places. So over here, we know that if you want to calculate any percentage change, then you have to do new minus old over old. What does that mean? You have to do the new value over here, which was 1.11. You have to do that minus the old value before the change, which was 1.28, and you have to divide over the old value again, which was 1.28, and you have to, of course, multiply by 100 to get the percentage. You're going to calculate that on your calculator, and you're going to get negative 13.28. So this is the percentage change that you will get. Double I says, using the information in table 2.1, explain the difference in the results between 0.6 mol negative cube decimeter and the 0.8 mol sucrose solutions. Use the term water potential in your answer. So we know that 0.8 mol has a greater percentage of the sucrose. That means that it has lower water potential, or it has less water than the one that has 0.6. So this means that the 0.8 concentration of sucrose will lead to higher amount of water being lost by osmosis. So you're going to say that the potato cube in 0.8 mol decimeter solution loses greater percentage mass of water loss. This is because there is a higher because it's going to move from high water potential to low water potential. This is because the water potential at 0.8 mol is less than 0.6 mol, and so the water potential gradient is greater in 0.8 mol decimeter cubed than 0.6 mol decimeter cubed. So we know that osmosis moves from high water potential to low water potential, and so when there is like a lower amount of water in some place, then all of the water will move to that place, and so this is the cause of the difference in results. Triple I, describe the expected appearance of a cell from a potato cube that has been immersed in distilled water for 30 minutes. So when you put a cell in the water for too long, it's going to become swollen and turgid. This is just like when you're holding a grape in your hand. It's if you just like put a little bit of pressure on it, it's going to pop. So this is like an example for the swollen and turgid. And also the vacuole is going to become larger. This is like the large empty space in the cell. This is the vacuole, this area. So it's going to become larger. So instead of being this size, it may become like this size. So this is going to increase in size as a result of having more water inside, and the cell contents will press on the cell membrane. B says, describe how the process of active transport differs from the process of osmosis. So active transport is very different from osmosis. Active transport is actually a process that uses energy, or ATP, from respiration. ATP means energy, it's the same thing. Movement is against a concentration gradient in the active transport, whereas in osmosis it is actually with a concentration gradient. So protein carriers are involved in the process of active transport. So this is the difference between the active transport and osmosis. Osmosis is a passive process, which means it does not use energy, which is the opposite of active transport. C says, state the type of plant cells that use active transport to absorb mineral ions from the environment. This is actually root hair cells that is in the roots of the plant. Now explain the effect of a lack of magnesium ions on the color of plant leaves. It will leave color changes from green to yellow. So this means that the plant or the leaf that was green, it will become yellow. As we all saw before in our lives, you have probably seen a leaf or a plant or some kind of flower, and it has like yellow leaves instead of green, and this is because it has a magnesium deficiency. It does not have enough magnesium, and magnesium helps with the production of chlorophyll or chloroplast that are used. It's the green pigment in the plant that is helping the plant with photosynthesis, so this is very important. 3a, question 3a, figure 3.1 is a photomicrograph of some cells lining the trachea. Describe the role of goblet cells. We know that goblet cells secrete mucus, which traps bacteria and pathogens. Explain how the cell labeled X in the figure 3.1 is adapted for its function. So over here, X, they are basically talking about this part over here, and it has a lot of like hair-like structures over here, this area. So they're talking about this area. Now it is ciliated and sweeps the mucus. So ciliated means that it has like those parts, those hair-like structures, and these actually sweep away the mucus. It's like sliding the mucus over the surface over here. So if there's something over here, it's going to be swept away. It's going to move towards this area. So this is sweeping. State the name of one other part of the body where the type of cell labeled X in figure 3.1 is found. This is actually the bronchioles. Table 3.1 contains some features of the breathing system. Complete table 3.1 to show the actions of each feature of the breathing system that occur to cause inspiration. So actually in inspiration, we know that the diaphragm contracts or it flattens, it becomes flat. So in the external intercostal muscles, they also contract, and the pressure in the thorax is decreased. So the external intercostal muscles are actually the opposite of the internal intercostal muscles. So if the external ones contract, then the other ones have to relax. So the ribs, they also move outwards and the volume of the thorax increases. C says that state the name of the gas that is excreted by the breathing system. This is actually carbon dioxide. Good ventilation is one feature of gas exchange surfaces. State two other features. They have to be one cell thick. Why do the good ventilation areas or gas exchange surfaces, why do they always have to be one cell thick? This is because the one cell thick, it provides a low diffusion distance or a small diffusion distance, so it will become faster for the diffusion to occur. So if there's something very thick, then it will take a lot of time for the air to move through it, unlike something that is thin. Large surface area, of course, for faster or more diffusion. State the name of gas exchange surface in humans. This is actually alveoli in our lungs. 4a, figure 4.1 shows the effect of light intensity on the rate of photosynthesis at different temperatures and concentrations of carbon dioxide. Describe and explain the reasons for the shape of lines B and C in figure 4.1. So actually, over here we can see they said describe and explain. So you have to describe, you have to see, you have to tell them what you notice about the picture, and also you have to explain why this occurs. So over here I can see that in C, it actually contains a much lower amount of carbon dioxide compared to A and B, and also over here it has a lower amount of light intensity. So there is the lowest rate of photosynthesis in C because it has lower light intensity, and also it has a low amount of carbon dioxide. So this is basically the limiting factor. And A, it has the highest amount of rate of photosynthesis over here. This is because it has the highest temperature. B and C have low temperatures, and it also has high carbon dioxide, and also it has a high light intensity. So this is the things that are needed for photosynthesis. These are actually the raw materials. So this will cause the rate to be very high. Now let's see. In B and C, the light intensity increases, the rate of photosynthesis increases, and then remains constant. So over here we can see that the graph, it was increasing and then it became suddenly constant. It became like a line, a straight line. This means that in the beginning, when it was increasing over here in this area, then it was actually a limiting factor. A limiting factor is something that is missing from the plant that is needed for photosynthesis. So for example, over here the limiting factor is carbon dioxide. There is low carbon dioxide. If we increase the carbon dioxide, then the rate will increase a little bit. So over here we have to also say that the light is a source of energy for photosynthesis. When the line increases, as I said, the light was a limiting factor. When the line remains constant, the light is no longer a limiting factor. For carbon dioxide, C has the lowest concentration, as I said, which is low compared to A and B, because A and B have 0.4%, and this one, C, has 0.04%. So this is much less than A and B. Also, carbon dioxide is needed, or it is a raw material for photosynthesis or reactant. At C, the carbon dioxide is the limiting factor at low light intensity. So this means that in C there was low carbon dioxide, so this was actually the limiting factor, and also there was a low light intensity. It had lower amount of light than B and A. And in B there was the carbon dioxide, it was a limiting factor at higher light intensity. In B, temperature is the limiting factor at high light intensity. So B actually has a limiting factor, which is the temperature, as I said. A has 25 degrees and B only has 15, so there is a difference, and it causes a difference also in the rate. It increases over here, and in B it decreases because of the low temperature. So this is basically all you need to say. B says C6H12O6 is one of the products of photosynthesis. This is actually glucose. State the chemical formula of the other product. This is actually oxygen, O2. You only have to write it down as O2. You can't write it down as oxygen in like a word like that. You have to write it down in its chemical formula, because if you write it down as oxygen like this as letters, then you're going to get 0 marks. Outline how the carbohydrates made during photosynthesis are used in plants. So of course, glucose has a lot of uses in the plant. It may be used to provide energy by respiration, and also glucose may be converted to sucrose. Sucrose is actually used in translocation, and it is used to provide energy to sinks. And also the glucose can be converted to starch, which is used as an energy storage. And glucose can be used as nectar. Nectar is used to attract insects for pollination. And glucose can be converted to lignin for cell walls. So this is also, there's a lot more uses, but this is mainly the uses that mainly come. 5a, figure 5.1 shows the stages involved in the protein synthesis. State the names of the parts labeled X, Y, and Z in figure 5.1. So actually X is the nucleus and Y is the mRNA. We can see that in this part is the nucleus. We know that these are the nuclear membranes, and the thing that is coming out over here, it is actually the mRNA, messenger RNA. And Z is the ribosome, where the mRNA where the proteins are basically made. So double I state what determines the sequence of the amino acids in the protein that is produced. Actually, this is according to the sequence of bases on the mRNA. So it's basically asking what makes the sequence of the amino acids in the protein like any different, or like what determines them. This is actually the order of the bases on the mRNA. And of course it is different for each type of protein. It's specific for each type of protein, I mean. So this is the answer. Now triple I, explain why the sequence of amino acids is important in the production of receptors molecules for neurotransmitters. The sequence of amino acids determine the shape of the protein. So for example, if you change the sequence of the amino acids just like a letter, then you're going to get a completely different shape of protein. So also the complementary shape is needed for receptor cells to bind with the neurotransmitter. You can think of it like phone numbers. Like if you actually mess up one number in your phone number, then you're going to get a completely different phone number, and it may belong to another person. So this is the same thing in the protein and the mRNA situation. B says, explain why body cells can have different specialized functions even though they contain the same genes. This is because not all the genes are expressed or switched on. So for example, in your, let's say, in your liver, only some types of genes are expressed. Not all of the genes are expressed. So the liver cells has all your genes, but only the liver genes are expressed. So only the ones that your liver needs are like expressed, or they are turned on, as cells only produce the specific type of protein they need. So they don't need the other ones, they only use what they want. Allele frequency in a population can be changed by natural selection and artificial selection. State the meaning of the term allele. It is the alternative form of a gene. So D says, describe how artificial selection differs from natural selection. In artificial selection, the humans actually control the selection, not the nature. So humans will choose the desirable features, which means that, for example, if they have a cow, they only want the cows that produce a lot of milk. They don't want the cows that don't produce a lot of milk. So what they will do, they will actually choose the offspring or the specific cows that actually have the features that they want, and they will allow them to reproduce. Only those ones will reproduce, and then the humans will control the selection. As a result, the genetic diversity is reduced, or the genetic pool. So this means that they will keep on reproducing until the offspring is what they all want, the cows that produce a lot of milk, and the other cows are not around anymore. So this is the humans artificial selection, and artificial selection can occur for economic reasons or aesthetic reasons. So what does that mean? Economic reasons, it is of course much more profitable for the farmer to have a cow that will produce a lot of milk. And also for aesthetic reasons, for example, they may allow like let's say specific, like they only will allow a cat that looks a specific way to reproduce, so that all cats will look that way, for example. That is for how it looks, like reasons, for aesthetic reasons. So E says mutation causes formation of new alleles, which increases genetic variation. State two other sources of genetic variation in populations. This is actually random fertilization or random mating and meiosis. You can also say mutation actually for this answer. 6a, a scientist monitored the changes in the pH in the muscles before, during, and after 2 minutes of vigorous exercise. The changes in pH are caused by the production of lactic acid. Over here we can see that after 2 minutes of vigorous exercise, the pH in the muscles started to increase very rapidly until it became constant at around 35 minutes. So let's see, complete the sentences to describe and explain the results in figure 6.1. The pH decreases from 7.07 to 6.55 during the vigorous exercise. So over here we can see it was actually 7.07 over here. This is 7.05, and this is 7.6, and this is 7.07. And then it decreased from that until it decreased to this part, which is 6.55. Now let's see the rest of the questions. There is not enough oxygenated blood supplied to the muscles. So this is the problem with oxygen debt. Oxygen debt is caused because the oxygen in the blood runs out. So then the body will go and respire anaerobically, which means it will make respiration without using oxygen. It will produce lactic acid, of course, and lactic acid cannot build up in the muscles. It will cause an oxygen debt, and also it can be very painful for the muscles. It takes 31.2 minutes for the muscle pH to return to its initial level after exercise. So actually I said that it took from 2 minutes until like something over here 35 minutes or something to return to its normal levels, which was 7.07. You can say anything between 31 to 32 minutes because it may actually differ from one person to another. pH value increases after vigorous exercise has ended as lactic acid is transported in the bloodstream to the liver. So during this time, the breathing rate and the heart rate may remain high. This is because the breathing rate and the heart rate remain high to provide oxygen. Like it causes your lungs will work much harder to allow more oxygen to enter, because there is basically like not enough oxygen in your blood, so it has to restore those oxygen levels. Yeast can respire anaerobically. Complete the balanced chemical equation for the anaerobic respiration in yeast. Actually, this is the equation. It's going to produce from glucose, it's going to produce alcohol or ethanol, which is this formula, and then it will produce carbon dioxide. Yeast belongs to the kingdom fungus. State one cell component that is present in the yeast cells but is absent in the animal cells. So yeast cells actually have cell walls, and they have cell vacuoles and plasmids. These are all things that are not found in animal cells. Thank you all for watching. This was the end of our exam. Make sure to subscribe and like so you can receive more content like this. And I will try to post every Sunday from now on. And I apologize if I don't post as frequently before. This is because I was really busy, and I apologize to all of you. And stay safe, everyone, and good luck with your exams.